Welcome to the Biochemistry Clinic. And today we look at pyruvate metabolism, which simply talks about how pyruvate is converted to glucose and also how glucose is converted to pyruvate. So mainly we know, after looking at the 10 steps of glycolysis, we saw how glucose is converted to pyruvate. But you have to know also that pyruvate can still be converted back to glucose using gluconeogenesis. Now, this is what you have to know. So glycolysis converts glucose to pyruvate. Pyruvate can still be converted back to glucose using gluconeogenesis. Now, what I want us to focus on today is the fate of pyruvate that is produced in glycolysis. What really happens to pyruvate after its production in glycolysis? Well, as you can see from this diagram, quite a number of things can happen. Pyruvate can go there, pyruvate can go there, pyruvate can go there and there. So I've created 4 different options for pyruvate after glycolysis. And I want you to pay particular attention to this. Now, the first option that I want to discuss is the conversion of pyruvate to alanine. Now, we know that carbon atoms are involved here. Like pyruvate is made of carbon atoms. So there are times when the body needs availability of carbon atoms. Pyruvate may not be able to move, for instance, from the muscle to the liver cells. So in order to allow pyruvate to also be stationed in the liver cells, for instance, pyruvate can be converted to alanine using the enzyme alanine transaminase. And this alanine is the one that is able to move from the muscles to the liver, for instance. And once it gets to the liver, it can still be converted to pyruvate. That pyruvate in the liver can still be converted back to glucose using this process that we're talking about, which is gluconeogenesis. So basically, this cycle is known as the Cahill cycle. Very interesting interplay, how pyruvate is converted to alanine simply for transport purposes. Once it gets to the liver, for instance, from the muscles, it is converted back to pyruvate, so the liver can utilize pyruvate. So if pyruvate is needed back into the muscles, it can first of all be converted to glucose using gluconeogenesis, and then the glucose will move from the liver and cross over to the muscles. So that's basically one route that pyruvate can take after it has been produced. So take note. Now, the other option is the conversion of pyruvate to lactate or lactic acid. Now, this is basically anaerobic glycolysis, meaning that glycolysis is taking place in the absence of oxygen. So you actually form lactic acid or lactic acid using lactate dehydrogenase. So this is a very, very critical aspect, and it is observed during infection. If we have to look at a few medical aspects there, during hypoxia and heart failure as well. So the lactic acid aspect has a lot of things to talk about. So for now, what you should know is that in the absence of oxygen, pyruvate can actually be converted to lactic acid using lactate dehydrogenase. So the other thing that you have to know is that this process will happen in the cytosol of the cell. So pyruvate to alanine and pyruvate to lactic acid both take place in the cytosol. So we move down here in the mitochondria. Pyruvate can be converted to oxaloacetate using pyruvate carboxylase, which simply means the addition of carbon to pyruvate to form oxaloacetate. Now, this oxaloacetate, as we may know, could be very important in the TCA cycle because it's oxaloacetate that combines with acetyl-CoA in the TCA cycle to form citrate in the first step. On the other hand, this oxaloacetate can be utilized in gluconeogenesis. We know that this can be converted to malate and shuttled into the cytosol where malate can be converted back to oxaloacetate and then proceed in gluconeogenesis. So this is one other possible option. Then the final and most critical, I think, is the decarboxylation of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex. Now, this is a very crucial route because it happens when you have sufficient oxygen, obviously. You produce acetyl-CoA in the mitochondria, and this acetyl-CoA will combine with oxaloacetate in step 1 of the TCA cycle and produce citrate. And then the cycle goes on to produce the likes of NADH, FADH2, GTP, and carbon dioxide. So basically, if you're asked to talk about the fate of pyruvate that is produced from glycolysis, please make sure you make mention of at least these 4 routes that pyruvate can take. Its conversion to alanine when the body actually needs carbon atoms, its conversion to lactic acid in the absence of oxygen, its conversion to oxaloacetate when there's need for oxaloacetate in the TCA cycle, or in gluconeogenesis. We know how this happens. This is converted to malate, and malate shuttled into the cytosol. And also, we need to talk also about how pyruvate is decarboxylated to acetyl-CoA into the TCA cycle. So basically, that's what I wanted you to know for today. And pyruvate metabolism. Please make sure you do not miss the next lecture, which addresses the TCA cycle reactions.